To our department head, Lieutenant Colonel Nathaniel P. Mendez, sir, to our training staff, to my senior and junior residents and interns, a pleasant morning to all of you. I am P2LT Redson Iro L. Valencia Medical Corps, second year resident from the Department of Orthopedics and Traumatology. And for this morning, I was tasked to report about the skeletal muscles. For the next 15 minutes, we will talk about the basic structure of the skeletal muscle, excitation-contraction coupling, and different types of skeletal muscle fibers. We can trace back the embryonic origin of the musculoskeletal system from the mesoderm, in which the lateral mesodermal plate will give rise to your bones and your connective tissues, while your somatic mesoderm will give rise to the skeletal muscle tissues. Skeletal muscle comes in different shapes and sizes, but the main structure of the skeletal muscle cell is basically the same. If we were to take a cross-section of a single muscle, we would find it is covered in a protective sheet of layer called your epimysium. The epimysium protects the muscle from friction against other muscles and bones. It also continues at the end of the muscle to form, along with other connective tissues, the tendon, which attaches the muscle to the bone. Each muscle is made up of a number of bundles of muscle fibers called your fasciculi, which are contained in another connective tissue called your perimysium. Fascicle is the smallest unit of structure visible to the naked eye. Each fascicle is composed of 10 to 100 muscle fibers. For strong large muscles such as your quadriceps, it has higher number of muscle fiber per bundle compared to smaller muscles for precision movements such as those in the hand that contains lower number of muscle fibers per bundle. Fasciculi are contained in another connective tissue called your perimysium, in which arterioles penetrate obliquely or at right angles to the muscle fibers and then run parallel. Mature cells in adult skeletal muscles are called muscle fibers or myotubes. They are differentiated multinucleated cells formed by the cytoplasmic fusion of immature mononucleated myoblasts. Each muscle fiber is also covered by a fibrous connective tissue called the endomysium. Beneath the endomysium is the sarcolemma, which is the muscle cell membrane, containing the sarcoplasm, a gelatinous fluid containing fats and glycogen for energy, and other organelles such as mitochondria for energy production. Each muscle fiber itself contains cylindrical organelles known as myofibrils, which are itself bundles of proteins, myofilaments, actin, and myosin, which are important in muscle contraction. Surrounding the myofibrils are a network of tubules and sarcoplasmic reticulum. T-tubules are invaginations of the outer membrane of each muscle fiber and functions to carry the depolarization of the surface membrane deep inside the muscle fiber. On the other hand, sarcoplasmic reticulum are membranous sacs that serves as a repository for calcium. Each myofibril can be broken down into repeated segments called sarcomeres, which are functional contractile units composed of myofilaments, actin, and myosin. Under microscopy, sarcomere form bands. The A band represents the myosin filaments. The I band represents actin filaments in adjacent sarcomeres where there is no overlap between the myosin filaments. The H band or H zone corresponds to myosin filament segment where there are no interdigitating actin filaments. The M line represents connections between adjacent myofilaments, and the Z disc, or your Z line, represents attachments of adjacent sarcomeres. Actin is a globular protein that is the chief constituent of the thin filament of the sarcomere. Other proteins are your tropomyosin and troponin. Tropomyosin blocks the binding sites of the myosin head. In this figure, you can see the tropomyosin blocking the site of the myosin head of the actin. Troponin C binds with calcium and counteracts the inhibitory effect of your troponin I, while your troponin T assists troponin C in binding to tropomyosin. Myosin have 6 distinct subunits, 2 heavy and 4 light. Heavy chains have 2 distinct parts, your S1 segments or your cross bridge that articulates with actin, and the S2 segments which forms a flexible neck which moves to allow articulation of the S1 segment. Before we talk about how the muscles contract, there are important concepts to remember. A motor unit is a structure in which all muscle fibers innervated by the same motor neuron. The size of action potential is directly proportional to the number of muscle fibers innervated. The force of contraction is directly proportional to the cross-sectional area, meaning larger muscles have greater force of contraction. And speed of contraction is directly proportional to the length of the muscle. Longer muscles have faster contractions. So how does the muscle contract? A action potential reaches the terminal axon in which it will stimulate release of acetylcholine in these vesicles. The acetylcholine in the synaptic cleft will bind to your acetylcholine-gated channels, giving in effect to sodium influx, hence depolarization. If there's enough depolarization, an action potential will produce and propagates deep into the central muscle fibers with the help of your T-tubules. Calcium will be released from the sarcoplasmic reticulum. Calcium will then bind to your troponin C, and troponin C displaces troponin I from the tropomyosin-actin complex. Troponin T will assist the binding of your troponin C to the tropomyosin. Then tropomyosin undergoes conformational change to allow myosin head engagement. Then myosin head engages and force generation occurs due to S1 segment rotation. Now let's talk about the different muscle fiber types. Muscle fibers in humans are of 3 main types. Type differs from each other in their size, speed, and resistance to fatigue. These characteristics are a function of their relatively aerobic or anaerobic metabolism. The pattern of activity is imposed on the muscle is the single most important factor in the fiber type expression. In such case, athletes who train extensively for endurance can reach proportions of up to 80% of type 1 fibers in their muscles. So let's proceed. Type 1 muscle fibers are slow fibers. They have large concentration of myoglobin, oxidative, and very fatigue resistance, hence meant for long distance running. Type 2 is subdivided into 2A and 2B. They are both fast fibers, however, Type 2A is both oxidative and glycolytic and fatigue resistance, while Type 2B is glycolytic and very fatigable, usually meant for sprinting or short bursts of contractions. Short, strong bursts of contractions. So in summary, we've talked about the structure of the muscles, especially the skeletal muscle. We've also discussed how does the action potential from the nerve propagates to the muscle fibers, eventually leading to its contraction. And we've also talked about the 3 types of skeletal muscle fibers. For the clinical correlates, in relation to our clinical practice, skeletal muscles are involved in deforming forces when it comes to fractures. In this example, we can appreciate that the proximal fragment of the left clavicle is pulled upward by your sternocleidomastoid. In joint dislocations, the skeletal muscles surrounding the dislocated joint are spastic, and in order to stabilize and lessen the pain, hence we need some muscle relaxant before we reduce the joints. Another importance of the skeletal muscle is that the surrounding muscular envelope provides a source of progenitor cells to the fracture callus and called satellite cells. These satellite cells have a role in the signaling and upregulation of the BMPs and IGF-1 and FGF-2 pathways typically observed in response to fracture. Hence, it's important to do only what's necessary, especially when dissecting the soft tissues. In doing the BRIDMA, in which we should not let any necrotic tissue to be left in the site of the injury, we can evaluate the viability of muscles by inspecting the color, capacity to bleed, contractility, and consistency. This figure is a leg of a 54-year-old male politician who sustained explosive injury last election. We can appreciate that the muscle is already dull red in color with apparently no appreciable bleeding. It doesn't also contract when stimulated, and the consistency was not very firm. Hence, we did an emergency BKA on this patient. So that's all. Thank you.